In this lesson, we will focus on what happens during glycolysis and what comes up when the process of glycolysis ends. And actually, glycolysis is the first stage in the process of cellular respiration, and it occurs in the cytoplasm of the cell. The word glycolysis means sugar splitting, which actually explains the whole procedure of glycolysis. And in this process, the glycolysis, which is 6-carbon molecule or 6-carbon sugar molecule, it is broken down into 3-carbon sugar molecule, and it is then oxidized and rearranged to form the molecules of pyruate, or 2 molecules of pyruate are produced. And it should be noted here that pyruate is the ionized form of pyruvic acid. Now we will see the overview of glycolysis. There are actually 3 steps to explain the overview of glycolysis. Number 1 is the priming step. You can see here that the ATP is giving its phosphate to the 6-carbon glucose molecule, and it is being converted to 6-carbon sugar diphosphate. So 2 phosphate groups are attached on 6-carbon sugar diphosphate, and it is called as priming. And then there is another step which is known as cleavage step or cleavage reaction. During these reactions, you can see that 6-carbon sugar diphosphate is cleaved into 2 3-carbon sugar phosphate. So in the next step, which is the energy harvesting step, NADH is formed and ATP is also formed in this process. And you can see here that NAD is being converted to NADH and energy is being harvested in this series of reaction. Now we will go into the detail of the process of glycolysis, which actually comprises of 2 steps. Number 1 is a preparatory step or preparatory stage, and it is actually the transfer of phosphate group from ATP to glucose. So as a result, a molecule of glucose 6-phosphate is formed, and an enzyme catalyzes the conversion of glucose 6-phosphate to its isomer, which is fructose 6-phosphate. You can see here the fructose 6-phosphate is being shown, and then at this stage another ATP molecule transfer a 2nd phosphate group, and the product is fructose 1,6 diphosphate or fructose 1,6 bisphosphate. And the next step in glycolysis is the enzymatic splitting of fructose 1,6 bisphosphate into 2 fragments. So each of these molecules contain 3 carbon atom. One is called 3-phosphoglyceraldehyde 3-PGL or glyceraldehyde 3-phosphate or G3P, while the other is dihydroxyacetone phosphate, and these 2 molecules are isomers, and in fact they are readily interconverted by yet another enzyme of glycolysis. So this was the first stage, and then there is a second stage or second step, which is an energy conserving step, and it is also called as oxidative or payoff stage. You can see here that this is very crucial stage in this process. 2 electrons or hydrogen atoms are being removed from a molecule of 3-PGAL, and it is being transferred to the molecule of NAD. This is of course an oxidation reduction reaction, with the PGL being oxidized and NAD being reduced. So during this reaction, a 2nd phosphate group is donated to the molecule from inorganic phosphate, which is present in the cell, and the resulting molecule is called 1,3-bisphosphoglycerate. So you can notice here that in your book there was written 3-phosphoglycerate, and I am showing you the figure with 3-phosphoglyceric acid. So the difference is, I mentioned earlier, that these terms which are mentioned in the book are ionized form of these acids. So both are correct, but you have to follow the pattern shown in the book. I have shown you this figure in order to show the carbon number, which are explained here better. So the oxidation of PGAL is an energy yielding process. There's a high energy phosphate bond is created in this molecule. At the very step of glycolysis, this phosphate group is transferred to a molecule of ADP, converting it into ATP, and the end product of this reaction is 3-PG. In the next step, 3-PG is converted to 2-PG, and from 2-PG a molecule of water is removed, and the product is PEP or phosphoenol pyruvate. Pyruvate then gives up its energy phosphate to convert a second molecule of ADP to ATP. The product is pyruate, or which is the ionized form of pyruvic acid. So this was all about the process. So we have to observe here that there is a need to recycle NAD. So why do we have to recycle the NAD? Because the NAD pool depletes when the reaction goes on again and again, and there is a shortage of NAD, and NAD needs another electron acceptor to accept electrons from NADH, so that NAD pool should not deplete, and it should be replenished. So some other molecules shall accept the hydrogen molecule which is attached to the NAD molecule, forming it to NADH molecule, so that the process of glycolysis can go again and again. So there are two pathways by which this procedure can then proceed on. So it can be either aerobic respiration, or it can be fermentation process, and fermentation can be the formation of lactate as a result, or ethanol. And in case of aerobic respiration, oxygen is the terminal electron acceptor, and in this case, after the stage of glycolysis, the pyruvate goes on to the Krebs cycle, and this is the normal process. And after the pyruvate is formed, normal process is, if oxygen is available there, then it undergoes the aerobic respiration process and proceed the process of citric acid cycle, and then the process of electron transport chain or respiratory chain. Now we will see the output of glycolysis, that what was the input and what was the output of the glycolysis. You can see here that first of all 2 ATP molecules were used in the first step or the preparatory stage, and then 4 ATP molecules were formed, and in addition 2 NADH molecules were also formed. So the formation of 2 ATP and 2 NADH, which are the electron acceptors, and they are carrying the electrons, and this is the net output in the process of glycolysis.